Hi everyone, we're going to do a quick demonstration of our enzyme reaction lab here. And today you're going to be working with hydrogen peroxide being decomposed and the enzyme catalase, which you're going to get from a banana. So in part A, we need some banana pieces to start with. So we're going to slice off, zoom in a little bit here, a little bit less than an inch, so about 2 centimeter piece of banana. And you're going to cut that slice into 4 pieces, okay? This first set of bananas is going to prep for later for part C, and so what I want to do with that is either put it in the freezer, or if you don't have a freezer accessible, some ice water will work also. So you're going to take all 4 pieces and chill them. And basically what we're doing is we're taking our enzyme and making it really cold. So either put it in ice water, or I'm going to take this and put it in the freezer. All right, now that that's done, I want another slice to use for part A here. So again, cut it up, and you're actually going to cut up a bunch of banana pieces so that you have a total of 12 for the whole day. All right, lots of banana. Set it aside, and every time you need a piece, you can grab one of those. The size is not crucial. We just want small pieces. You could even make yours smaller than mine. So now you're going to need another container. Make sure that throughout the lab experiment you have 3 identical containers, something consistent. For the other containers you can use, whatever you can find around the house. So I'm going to use a random container, and I want to soak my 3 banana pieces, soak 2 banana pieces in room temperature water. So a couple pieces of banana, soak them in room temperature water, just enough to cover them, and I'm going to set those aside now. All right, now we need containers for our bananas to react in. So 3 cups work well. You can label those cups with some tape or a piece of paper, but I've got 3 cups, okay? And if you don't have cups as a backup, you could also use a plate, where maybe on your plate you're going to have area 1, area 2, area 3, or keep track of it. But containers are definitely better. And another type of container that works actually better than a big cup even is if you happen to have something small. This is a shot glass, but something small, that works just fine. It means you need less liquid when you do the experiment. So I've got 3 cups here, but something small or a plate if you need to will also work. All right, so now I've got my 3 cups and my bananas. I'm going to take banana into cup 1 and banana into cup 2, and then no banana into cup 3. That means there's no enzyme in cup 3. And then I'm going to follow my directions. I'm going to barely cover this banana with hydrogen peroxide. Okay, this is the substrate in these reactions. So barely cover it so you don't run out of peroxide. Into the second container, you're going to do the same amount, but of this is just tap water. And then in the third one, I'm going to put peroxide, but with no banana, okay? So we would call this a positive control for what we expect to happen. And then we would also think of the other 2 as negative controls. Negative control. So what happens if I don't have any substrate? And what happens if I don't have any enzyme is what you're going to be looking for. So you'll take a look at those, observe them for 10 minutes. You should see bubbles forming around your banana. Those bubbles are oxygen bubbles, and that is evidence of a reaction happening. If you do not see bubbles, that means you don't have a reaction, and probably you don't have a reaction because you're missing either the enzyme or the substrate or both. Okay, so that's our basic setup for part A. You're going to dump everything out, clean it up, and be ready for part B now. All right, in part B, we're going to be looking at the pH influence. So we're going to have some water, which is neutral. If you have pH paper, use it at this point to check your pHs as we work through this part. If you don't, that's fine, just estimate. Baking soda will be basic, and then we'll have vinegar, which is acidic, going into our 3rd cup. But first we want to pre-treat our enzyme, so pre-treat our banana. And so you're going to have a total of 6 containers here, okay? And you can just collect whatever containers you have around for your pre-soaks. So into each of those pre-soak containers, you want to take one of those banana pieces that you cut up earlier, okay? And we want to do the same thing to all of them. We want to soak all of them. So I'm going to soak this first one in water. And I actually went out of order here. So with the baking soda one, before you have that banana in there, you want to put about a teaspoon of baking soda and dissolve it in some water. An estimate's fine. So I've got a teaspoon of baking soda, add some water. If you remember from our acid base lab, if you've done that, baking soda is slightly basic. So I've got some baking soda. Now I can drop that banana back in there to get a basic soak. And then in the third one, I want to soak in an acidic pH. So I'm going to pour some vinegar, just enough to cover my little banana there. Okay. And if you don't have much, say, vinegar or peroxide, be working with the smallest containers you can today. So we're soaking in neutral water, soaking in baking soda, soaking in acid. You're going to let those soak for 5 minutes. And while you wait, you want to have your 3 clean cups, okay? Containers, cups. Into each cup, hopefully you paid attention to how much peroxide you used last time. But we want just enough that when I move that banana over, it'll be able to be submerged. So I'm going to put some peroxide into each of the 3 cups, enough that that banana will be submerged when I put it in. And so what you're going to do after 5 minutes have passed is you will come along with a cleaned off fork each time, and you'll take that banana out of its soak and put it into the peroxide and see what happens. Okay, then we'll take our banana that came from a base, put it into peroxide, see what happens. I didn't put enough peroxide in these, but clean off your fork from the acid, put it in the peroxide, see what happens. Okay, again, if you see bubbles forming, that's going to be indicating that you have a reaction. Note, do the bubbles form fast? Do they form slow? Do they form not at all? So that's how part B is going to work. All right, on to part C. Everything's been cleaned out. I've got my same 3 cups and I have 2 containers now. So if you read the directions for part C, you're going to have banana pieces ready, and then your 3rd banana piece is in the freezer or chilling in some ice water, okay? Freezer works better, but if you have to use ice water, that's fine too. So now we're gonna test what happens when I take these enzymes and I put them at different temperatures. How does temperature impact the enzyme, okay? So into container A, I'm gonna drop my banana and I'm gonna cover it with some water. So we're doing that pre-soak treatment. Into container B, I'm gonna put a banana chunk, and this banana chunk is gonna need to be covered with some hot water. So heat it up with a tea kettle or with in the microwave or whatever. So I've got some hot water here in my tea kettle, and I'm gonna pour that over this banana. We wanna be consistent in our experiments. So now we're gonna let all of this sit here and soak for 5 minutes at least. And then, like in the previous round, previous part, we're gonna take peroxide, put it into my cleaned cups here. And after my 5 minutes has passed, I will transfer each of these bananas into my cup with the peroxide, and I'll see what kind of reaction happens. Okay, into the hot water with that peroxide. Make sure you're, and you can grab your banana from the freezer if you've got it in the freezer, and so that goes in here as well. Like last time, I haven't fully submerged these bananas, but it's gonna work better if you have enough peroxide to submerge them. So for part D then, you're gonna take this same type of experiment, okay, and you're gonna come up with what you want to do, okay? So some ideas that you might do, you might say, well, that was interesting to use white vinegar. Would I see the same results if I use different kinds of juices? Or if I use different kinds of vinegars? You could even juice an orange or a lemon if you have something around in your house. You could also try different lengths of time on your temperature. So you've got your hot water, you've got your ice water. What happens if I chill it for 1 minute compared to 5 minutes? So we could do no chilling, 1 minute chilling, 10 minutes chilling, or no heating, 1 minute heating, 10 minutes heating. You could also take your, say, vinegar and dilute it. So that would be take like a 1/4 cup of this, add a 1/4 cup of water, and now you have less vinegar, and you could see, does a lower concentration of acid have the same impact on this reaction? So those are some ideas. Do the experiment. Not a bad idea to have something that's like consistent, which we would call our positive control, and then do a change and a different change, just like you've seen the other experiments set up. So have some fun with this, and I will look forward to seeing your results.